The Constitution of India, Preamble. We, the people of India, having solemnly resolved to constitute India into a sovereign, socialist, secular, democratic republic and to secure to all its citizens justice, social, economic and political, liberty of thought, expression, belief, faith and worship, equality of status and of opportunity, and to promote among them all fraternity, assuring the dignity of the individual and the unity and integrity of the nation. In our Constituent Assembly, this 26th day of November 1949, do hereby adopt, enact and give to ourselves this Constitution.
Hello my dear children, welcome back to the biology class. In the previous class, we had just started the cell division, mitosis, which leads to the growth of the body. And today we will be studying more about mitosis. Okay, so mitosis consists of two stages, isn't it? In the previous class I had told you that means the mitosis consists of the division of nucleus and the division of cytoplasm. The division of nucleus is known as karyokinesis and the division of cytoplasm is known as cytokinesis. So first the division of nucleus takes place, which is followed by the division of cytoplasm. Okay, so first we will learn karyokinesis, that is the division of nucleus. So karyokinesis completes with 4 stages. Karyokinesis consists of 4 stages and they are prophase, metaphase, anaphase and telophase. Okay, and the prophase, it consists of a beginning prophase and late prophase, early prophase and late prophase. Okay, so in the prophase, we know that when the cell attain maturity, all the cell organelle will be formed there. The quantity of cytoplasm will increase, the size of the cell also increases, and inside the cell, inside the nucleus, the parts of nucleus also we have learned. Chromatin Reticulum is present inside the nucleus, isn't it? The chromatin fibers, network of chromatin fibers. Okay, so I told you the prophase consists of 2 stages, that is early prophase or beginning of the prophase and the end of prophase or late prophase. Okay, so in the beginning of the prophase, this chromatin reticulum, they condenses to form chromosomes. That is happening in the beginning of the chromosome. Chromatin reticulum means network of chromatin fibers. They join together to form elongated or stranded chromosomes. And I will tell you one more thing. We can see the chromosomes in the nucleus only at the time of cell division. In all the other times, the chromosomes will be in the form of chromatin reticulum. Okay, so you can see in the early prophase or the beginning of at the beginning of the prophase, you can see the chromatin reticulum condenses to form chromosomes, and these chromosomes duplicates. Duplicates means, so when this chromatin reticulum condenses to form chromosomes, they will be having a single strand. They are single stranded. Okay, you can see here the structure of a chromosome. Okay, when they duplicate, what happens? No, they produce one more sister chromatids, one more strand. So they will become double stranded and both the strands are alike in their composition, in everything they are ഈക്വൽ ഒരേപോലെ ഒരേ ക്യാരക്ടറിസ്റ്റിക്സ് ആയിരിക്കും ഈ ക്രോമസോമിൻ്റെ രണ്ട് സ്ട്രാൻഡ്സിനും ഉണ്ടാവുക. ഓക്കെ, സോ ദ ഡ്യൂപ്ലിക്കേറ്റഡ് ക്രോമസോം കണ്ടെയ്ൻസ് 4 chromatids. Okay, so when it comes to the end of the prophase, the nuclear membrane and the nucleolus disappears, and already the chromosomes, the chromatin reticulum condenses to form chromosomes, and these chromosomes duplicates. Already it is there, and we have already learned the cell organelles, isn't it? So centrosome is also a cell organelle which is present in the animal cell, and the centrosome is seen very close to the nucleus, isn't it? And you have studied that the centrosome play a major role in the cell division, isn't it? So the centrosome consists of 2 centrioles, and these 2 centrioles, when this at the time of cell division, the centrosome divides, and these 2 centrioles released from the centrosome, and they move to the opposite poles of the cell. Okay, and spindle fibers are arised from the centrioles. Spindle fibers are small micro protein filaments. They are formed from the centrioles. So these are the changes takes place in the nucleus during the prophase. Did you understand, all of you? So let us watch it. So you can see here in the prophase, the chromatin fibers condenses to form elongated chromosomes. Okay, did you see? These are the elongated chromosomes. Okay, and these chromosomes are the duplicated chromosomes, as they are double stranded. So these 2 are the chromatids, and this center part is known as the centromere of the chromosome. Okay, and I told you at the end of the prophase, you can see here the nuclear membrane disappears, nucleolus disappears. Okay, and the centrioles move to the opposite poles. These 2 are the centrioles, and the centrioles move to the opposite poles of the cell. So these are the changes that happens during the prophase. I think you have understood. So the first thing is the chromatin reticulum condenses to chromosomes. Then the chromosome duplicates എന്ന് പറഞ്ഞാൽ ആദ്യം കണ്ടൻസ് ചെയ്ത് ക്രോമസോം ഉണ്ടാകുമ്പോൾ അത് സിംഗിൾ സ്ട്രാൻഡഡ് ക്രോമസോം ആയിരിക്കും. വെൻ ദേ ഡ്യൂപ്ലിക്കേറ്റ് ദേ പ്രൊഡ്യൂസ് 1 more സിസ്റ്റർ സ്ട്രാൻഡ് അല്ലെങ്കിൽ സിസ്റ്റർ ക്രോമാറ്റിഡ്സ്. അപ്പോൾ 2 എന്താ പറയുക സ്ട്രാൻഡ്സ് ഉണ്ടാവും ഒരു ക്രോമസോമിൽ. അതിനെയാണ് ഡ്യൂപ്ലിക്കേറ്റഡ് ക്രോമസോം എന്ന് പറയുക. Then the next stage is the nuclear membrane and nucleolus disappear, and the centrioles are formed from centrosome, and they move to the opposite poles, and the spindle fibers are formed from the centrioles. So these are the changes that take place in the nucleus during prophase. Hope you all have understood. Now we are moving on to the second stage known as metaphase. Okay, during the metaphase, the chromosomes, they aligned at the equator of the cell. You can see here they are aligned at the equator or the center of the cell, and the spindle fibers that are formed from the centrioles connected at the centromere of each chromosome. You can see it very clearly. The spindle fibers formed from the centrioles connected to the centromere of each chromosome, and you can see very clearly here the chromosomes are aligned or they are arranged at the center of the cell, and this is a significant characteristic of metaphase. When we are asked to find out the stages of karyokinesis, very easily we can identify the metaphase, because only in the metaphase we can see the chromosomes they are aligned or arranged at the center of the chromosome. So this is a significant characteristic of metaphase. Okay, and the spindle fibers which are formed from the centrioles are connected to the centromere of each chromosome. So these are the changes that take place during the metaphase, and the third stage of karyokinesis is anaphase, and in this phase we can see that the spindle fibers which are connected to the centromere pulls the chromatids. As a result, the chromatids get separated and the separated chromatids move to the opposite poles of the cell. As a result, daughter chromosomes are formed. Okay, let us see how it happens. You can see here this is the centromere, spindle fibers, they pull the chromatids. As a result, they separated and moved to the opposite poles of the cell. Okay, very clearly you can see here the spindle fibers separate the daughter chromosomes, and you can see here 4 daughter chromosomes are arranged at the opposite poles of the cell. Okay, so here in the separation of the chromatids, the spindle fibers play a major role. They are connected to centromere and they pull the chromatids. As a result, the chromatids separated and the daughter chromosome, 4 daughter chromosomes, 8 daughter chromosomes are there, isn't it? 4 daughter chromosomes move to one pole and another 4 daughter chromosomes move to the another pole. They move to the opposite poles of the cell. So altogether 8 daughter chromosomes are formed here. 4 at one pole and the 4 at another pole. This is what happens during anaphase. And the last phase is telophase, and during telophase what happens? The nuclear membrane reappear around the daughter chromosomes. Okay, and nucleolus, nucleoplasm, all these will form again in the chromosome. Centrosome also reappear in the near the nucleus, and 2 daughter nuclei are formed at the opposite poles of the cell, and the chromosomes, they again change into chromatin reticulum. At the end of the nuclear division, the chromosomes again change into chromatin reticulum. Did you understand? So now 2 daughter nuclei are formed. So this is how the nucleus divides. Okay, so let us watch what happens during the telophase. So during telophase, the formation of chromatin reticulum, and the nuclear membrane regenerates around the chromatin reticulum. Nucleolus and nucleoplasm also develop. As a result, 2 nuclei are formed with the same number of chromosomes as in the parent cell. Okay, so here there is no change in the number of chromosomes. At the beginning, what was the number of chromosomes? 4 chromosomes, isn't it? And now, what's the number of chromosomes in each nuclei? 4 chromosomes. Each nuclei consists of 4 chromosomes. Understood? So there is no change in the number of chromosomes. Okay, so this is the characteristic feature of mitosis. So now the nuclear division is over, and after the nuclear division, what happens? The existence cell also has to divide, then only 2 daughter cells will be formed, isn't it? So after the nuclear division, the cytoplasm will divide, and that is known as cytokinesis. Okay, so the nuclear division is more or less same in animals and plants. Okay, but the cytoplasmic division is quite different in plants and animals. It is different. It is not same in animal cell and plant cell. Okay, so let us see what are the changes or what is the difference in cytoplasmic division in the plant cell and animal cell. So first we will study the cytoplasmic division in animal cell. After that we will study the plant cell. So in animal cell, the cytoplasmic division is taking place by the formation of an invagination, or the plasma membrane invaginates at the center of the cell, or it invaginates between the two daughter nuclei. Understood? Invaginates means cleavage furrow is formed between two daughter nuclei in the plasma membrane, and this cleavage furrow deepens to the center. As a result, the cytoplasm divides, and as a result, two daughter cells are formed. So let us see that. Okay. So now you can see here a cleavage furrow, or this is known as the invagination. ഈ സെൽ മെംബ്രെയിൻ ഓർ പ്ലാസ്മ മെംബ്രെയിൻ ഇങ്ങനെ ഉള്ളിലേക്ക് ഇങ്ങനെ കുഴിഞ്ഞ് വന്ന് ഒരു വിടവ് ഉണ്ടാക്കുക, അതിനെയാണ് ഇൻവാജിനേഷൻ എന്ന് പറയുന്നത്. ഓക്കെ, സോ ഈ ക്ലീവേജ് ഫറോ നോക്കിക്കോളൂ. കണ്ടോ, ദിസ് ഈസ് ഹൗ ദ സൈറ്റോപ്ലാസം ഡിവൈഡ്സ് ആൻഡ് ടു daughter cells are formed, and this process is known as cytokinesis. Okay. So one mature cell or mother cell divides to form two daughter cells. Each daughter cell carries one nucleus. Okay, so this is how the nuclear division and cytoplasmic division takes place in the animal cell, and the cytoplasmic division is different in plant cell when compared to animal cell. Okay, so let us see how does cytoplasmic division take place in plant cell. In plant cell, small vesicles are formed like this between the two daughter nuclei. Okay, these are the small vesicles, and these small vesicles join to form a cell plate, and this cell plate to extends, അത് ഈ സെൽ പ്ലേറ്റ് ഇത് ഫോം ചെയ്തൊരു സെൽ പ്ലേറ്റ് ഉണ്ടാവും. This cell plate extends to both sides and joins with the plasma membrane. So this is the plasma membrane, and when this cell plate extends to both sides and join with the plasma membrane, the cytoplasm divides. As a result, two daughter cells are formed, and each daughter cells contains two nuclei also. നേരത്തെ ന്യൂക്ലിയർ ഡിവിഷനിൽ രണ്ട് ഡോട്ടർ ന്യൂക്ലിയർ ഉണ്ടായല്ലോ, അതിൽ ഓരോ ന്യൂക്ലിയസ് ഓരോ സെൽസിൽ അതിൻ്റെ സെല്ലിൻ്റെ ഓപ്പോസിറ്റ് പോൾസിലാണ് ഉണ്ടായിട്ടുണ്ടായിരുന്നത്. അപ്പോൾ സൈറ്റോപ്ലാസം ഡിവൈഡ് ചെയ്യുമ്പോൾ രണ്ട് സെല്ലുണ്ടാവും, ഓരോ സെല്ലിലും ഓരോ ന്യൂക്ലിയസ് വീതം ഉണ്ടാവും. Okay, so this is how the cytoplasmic division takes place in plant cell. Okay, so now you understood the difference between animal cell and plant cell in the case of cytoplasmic division. Okay, so during the mitosis, a mother cell divides to form two daughter cells, and each daughter cell grow and further divides, and during each division what happens? Before the division, not during the division, before the cell division, the genetic material, that is the chromosome, duplicates, isn't it? And as a result, there will not be any change in the number of chromosomes. അതായത് ഓരോ ഡിവിഷൻ നടക്കുമ്പോഴും ഈ ജനറ്റിക് മെറ്റീരിയൽ the genetic material duplicates. After that only the cell divides, and because of this there is no change in the number of chromosomes. അപ്പോൾ നമ്മൾ ചാപ്റ്ററിൻ്റെ ബിഗിനിങ്ങിൽ കണ്ടിട്ടുണ്ടായിരുന്നു അല്ലേ, ഒരു ബോർഡിൽ ഒരു ടീച്ചർ എഴുതിയിരുന്നത്, അതായത് ഒരു പാരൻറ്റ് സെൽ വിത്ത് ക്രോമസോം നമ്പർ 46, ആൻഡ് ദിസ് പാരൻറ്റ് സെൽ ഡിവൈഡ്സ് ടു ഫോം ടു ഡോട്ടർ സെൽസ്, ആൻഡ് ഈച്ച് ഡോട്ടർ സെൽ ക്യാരീസ് 46 chromosomes, which means even after the division there is no change in the number of chromosomes. and this occurs only because of the duplication of the genetic material that takes place before the cell division. Did you understand? So this is one of the significant characteristic of mitosis. So the cell divides several times, you know that. So even after several divisions, there will not be any change in the chromosome number. ഒരു സെല്ല് എത്ര പ്രാവശ്യം ഡിവൈഡ് ചെയ്തോട്ടെ, ഓരോ പ്രാവശ്യം ഡിവൈഡ് ചെയ്തിട്ടുണ്ടാവുന്ന ഡോട്ടർ സെൽസിലും അതിൻ്റെ പാരൻറ്റ് സെല്ലിൻ്റെ അതേ നമ്പർ ഓഫ് ക്രോമസോംസ് തന്നെയാണ് ഉണ്ടാവുക. എല്ലാ സെൽസിലും നമ്മുടെ ബോഡി സെൽസിലും ദ ക്രോമസോം നമ്പർ വിൽ ബി 46. Even after several division, there will not be any change. Okay, so that is one of the characteristic of mitosis. And you know that mitosis is a controlled division, which means one parent cell divides to form two daughter cells, isn't it? Then these two daughter cells divides to form 4 cells, then 4 cells divide to form 8, then 16, then 32, then 64, like that it goes, isn't it? So it's a controlled division. So when any disruption takes place in this controlled process, a cell divides excessively and proliferates, and the proliferation of that particular cell will take place. Proliferation എന്ന് പറഞ്ഞാൽ uncontrollably ആ സെല്ല് ഡിവൈഡ് ചെയ്യും, ആൻഡ് ദിസ് ലീഡ്സ് ടു ക്യാൻസർ. ഇങ്ങനെയുള്ള അൺകൺട്രോൾഡ് ഡിവിഷൻ അല്ലെങ്കിൽ പ്രോലിഫറേഷൻ ഓഫ് സെൽസ് ആണ് ക്യാൻസേഴ്സ് ഒക്കെ ഉണ്ടാവാനുള്ള കാരണം. ഓക്കെ, അപ്പോൾ സെൽ ഡിവിഷൻ എപ്പോഴും ഒരു കൺട്രോൾഡ് പ്രോസസ്സായിട്ട് പോയാൽ എവറിത്തിങ് വിൽ ബി നോർമൽ. ഓക്കെ, now let us see what are the significances of mitosis. ഒന്ന് ഞാൻ ഓൾറെഡി പറഞ്ഞു, the chromosome number remains constant even after several division. That is one of the significance. Then the next one is mitosis helps to repair the worn out tissues. അതേപോലെ തന്നെ it also helps in the growth of the body. അപ്പം നിങ്ങളൊക്കെ വളർന്ന് വലുതായില്ലേ, നിങ്ങൾ ഒരു 10 years ബാക്ക് നിങ്ങൾ എങ്ങനെയായിരുന്നു? അതുപോലെയാണോ നിങ്ങളിപ്പോൾ കാണുന്നത്? അല്ല, യു ആർ ഗ്രോയിങ്, isn't it? So the mitotic division helps in the growth. So these are the significances of mitosis. Okay, so hope you all have understood the nuclear division and cytoplasmic division. So watch the video. When you watch the video, keep the textbook with you. Okay, so after watching the video, if you have any doubt, you call me and clarify your doubts. Okay, so now I am winding up this class. See you in the next class. Until then, this is your Maya teacher signing off.